metabolic pathways. So how can we use enzymes to help catalyze reactions within a pathway? So first, let's look at and define what is a metabolic pathway. Well, a metabolic pathway is a series of linked reactions. So if we look at our figure over here, we can see that we have multiple reactions, right? We have 1, 2, 3, 4 reactions. And these reactions goes from a substrate being able to convert it to an end product. So examples of this type of reaction would be the ones found in photosynthesis, the ones found in cellular respiration, and so on, where the original substrate can be used and we go through these different reactions to end up with the end product. What you also can notice is that if we have an enzyme involved, an enzyme will actually bind to the substrate. So we can see the enzyme binding to the substrate over here. And this would allow a reaction to occur much faster than it would without the enzyme. So an enzyme kind of helps catalyze reactions so that the substrate can quickly go through these reactions quicker than it normally would without the enzyme in order for us to get an end product. Most enzymes are made of proteins. There are also cofactors that can work with enzymes, but basically these proteins will recognize and bind to a specific substrate, and it will bind here at its active site. So you can see over here, and once the substrate is at the active site, this can lead to a reaction and eventually leads to a product. The substrate is specific to the enzyme, meaning that not all substrate will bind onto any enzyme. The enzyme and the substrate are specific to one another, and it's through this interaction that we're able to catalyze. So by catalyze, we mean helping a reaction occur faster than it normally would without the enzyme. It's able to catalyze these different reactions. As we mentioned, the enzyme will have an active site that's specific to where the substrate will bind. The interaction of the substrate and enzyme to result in product, the enzyme does not get used up in the reaction, meaning that it's not created or destroyed. It is part of the reaction, but we don't actually use it up. So we don't actually, once we're done with the reaction and the enzyme has converted the substrate into the product, the enzyme just gets recycled again. So you end up with this enzyme over here that just gets recycled again and gets placed into the beginning again. Okay, each enzyme is specific to a particular reaction. Why is that? Well, that's because the substrate that interacts with that enzyme is also specific. We can think of this as the lock and key model, where we actually find that similar to how specific a lock and key is, where the key will only fit into certain locks and not all locks, we can consider that the key here being the substrate and the lock here being the enzyme. So you have to have the correct substrate in order to actually fit into that enzyme. So you can see here, this seems like it has a very particular shape that accommodates this substrate, right? We also have the induced fit model. This model, instead of saying there's an exact fit, suggests that the enzyme will actually undergo a shape change. So it kind of changes shape, and it changes this shape in order to accommodate the shape of the substrate itself. Upon the products being released after reaction occurs, the enzyme will then return back to its original shape. Again, it returns back to its original shape. The enzyme is not used up, and now the enzyme can be recycled to bind to another substrate and catalyze that reaction. Okay, so enzyme can be inhibited. So there are times in which we actually want to inhibit a reaction. It can be observed in a feedback inhibition in which we have too much products. And so because we have too much products, our body is kind of like, no, you don't need to make any more products. And so the product would then become the substrate and will bind onto the enzyme. So here you have the end product actually binding onto the enzyme. This leads to the enzyme changing its shape. And when it changes its shape, it can no longer bind onto substrate to push and catalyze that reaction anymore. An enzyme can also be inhibited by actually combining with certain substrate. So here we have this example where we have cytochrome oxidase. Cytochrome oxidase is an enzyme that catalyzes the final step of the electron transfer chain in respiratory cells. When this enzyme is exposed to cyanide, cyanide is a poison. Cyanide will actually bind on to this enzyme, preventing it from being able to catalyze that final step in the electron transfer chain. This is what results into nausea, vomiting, unconsciousness, and possibly death. Another example of this is penicillin. So bacteria that are exposed to penicillin can actually result in killing the bacteria because the penicillin will actually interact with certain bacterial enzymes to prevent the bacterial enzyme from functioning properly. So now that we've kind of introduced all this with regards to substrate and product, let's talk about activation energy. So what exactly does the enzyme do to catalyze a reaction? So without an enzyme, if we look over this plot right here, we can see that without an enzyme, the reaction would normally take a lot of energy, right? So here's the tip up here. It normally would take a lot of energy to push these substrate into these products. What an enzyme does is an enzyme will actually lower the amount of energy that's required. So it will actually lower the energy of activation that is needed in order to push these substrate into product. So basically, an enzyme affects the energy of activation, which is the energy that's needed to cause the molecule to react with one another, so the substrate to react with one another to make the product. And without the enzyme, we have this huge energy barrier, right? We have this large energy barrier right here that we must overcome in order for the reaction to occur. The enzyme makes that energy barrier slightly smaller. So with the enzyme, this energy barrier is here now, is a lot smaller compared to without the enzyme, and the reaction is able to occur faster. So let's answer this question right here. Which of the following is not true regarding enzymes? So taking a look at our graph on our picture here on the right, is it because they A, increase the amount of energy that's required for a reaction to run, B, they speed up the reaction, C, they possess an active site, D, that they are protein, and E, enzymes are not used up by the reaction. So take a moment and think which one is not true. Okay, I hope that you all said A is because they increase the amount of energy that's required for a reaction to run. This is false, right? An enzyme does not increase the amount of energy that's required to run the reaction. It actually lowers that activation energy and allows the reaction to run faster than it would without the enzyme. Okay, so we go back to the previous slide, right? Just recall that they're actually lowering that activation energy that's required to get over that hump to convert the substrate into product. Okay, and that's it for enzymes.